Good morning. So I don't have any Taylor Swift songs, but lots of self-doubts after Dr. Sear's excellent talk, who has clearly raised the bar. So thank you. I'm going to switch topics a little bit and talk about gene therapy for ARVC. We've heard a lot how important the genetics have become in the management, in the diagnosis and management of ARVC. And in the last couple of years, we put a lot of emphasis on diagnosing ARVC with genetics. And now I'm delighted that a new door has opened, a new exciting opportunity that we can use that genetic information to alter the course of the disease and potentially cure it. So I don't have any relevant disclosures, and I structured my talk today. It's a lot of feedback. Let's see. I hope that this is better. Good. I want to talk about three different things. So first, about what is gene therapy? What do we really understand when we talk about gene therapy? How does gene therapy work? And last, talk a little bit about gene therapy trials for ARVC. So what is gene therapy? So cells use the information contained in the DNA and the genes to carry out their functions. And Brittany told us that already this morning, that we have the instructions for the proteins that carry out the function in a cell and in an organism are encoded by the DNA, by the genes. And if everything goes well, you have the DNA encodes a protein, and ultimately you have a function that's carried out by that protein. If you have a change in the DNA, which we call a mutation or a pathogenic variant, that leads to an altered protein, or in worst case scenario, to a complete loss of the protein. So you have a reduced function or ultimately even a loss of function, and that will lead to disease. So gene therapies go back to the root cause. We know that there is a change in the genetic material, so can we use that and change the genetic material to treat or prevent a medical condition? And we use gene therapy to do a couple of things. So we can replace a gene that is causing a medical problem. We can repair a gene so that it no longer causes a medical problem. We can turn off a gene that is causing a medical problem. And we can add a gene that helps the body to fight a disease. Now, to simplify it, when I talk today about gene therapy, I'm talking mostly about gene replacement therapy. So to replace a gene that is defective and introduce that in the body. We're not going to talk about turn off a gene or gene editing. That would be a completely separate topic. Gene therapy has been around for the last 20, 30 years, and there are more than 3,500 gene therapy trials that have been conducted or that are actively ongoing. The vast majority of them actually deal with cancer. More than nearly 2/3 of them deal with cancer. But about 5% are also gene therapy trials in cardiovascular diseases. So what kind of conditions are we thinking about treating? There are hypertrophic cardiomyopathy, so when your heart muscle gets too thick, dilated cardiomyopathy, where the heart enlarges and becomes weaker, ARVC, and also heart failure that is associated with several muscular dystrophies. I think it's getting a lot of feedback. Let me see whether I can. Is there a way to turn this off? Thank you. And there are a lot of companies who are actually interested in pursuing gene therapy in cardiovascular diseases. I listed some of them here, and some of them are also have graciously agreed to sponsor today's seminar. So how does gene therapy actually work? So wouldn't it be nice if we can just take the DNA and inject it into patients? Well, actually, it's not the way it works. We have a very low efficiency if we introduce just naked DNA. So that really doesn't work. So there is a solution to that, and we know that viruses are really good at infecting humans and introducing their DNA into humans. So what if we can put the DNA of interest into a viral vector that we can use to transmit or transduce this DNA into humans? And the most suitable virus that has emerged over the last couple of years is called adeno-associated virus, or in short, AAV. So you'll hear me talk a lot about AAV today. And there are a lot of active gene therapy trials going on with AAVs. So you can use it to inject into the eyes if you have some disease of the retina or retinitis pigmentosa. There are some metabolic diseases where AAVs are used, central nervous diseases, neurodegenerative diseases where AAVs have been used successfully, and then also for muscular dystrophies, whether that's Duchenne muscular dystrophy or limb-girdle muscular dystrophy. And the most impressive results I have seen are in a disease called spinal muscular atrophy, or SMA, and I'm going to talk about that a little bit later. So what are the benefits of AAV? Well, it's, first of all, non-pathogenic. What does that mean? So infection with AAV is not associated with disease in humans, which is important if you want to use it for trials. It's replication deficient, so it can't replicate on its own. It creates a very low immune response. It does create an immune response, but a rather small one. And importantly, it leads to long-lasting gene expression. So you introduce a gene, and you can be sure that it's not gone within a month or two. Very importantly, when we consider gene therapy for the heart, AAV can efficiently infect both non-dividing and dividing cells. Human heart cells are what we call terminally differentiated, so they don't divide afterwards, and they're much harder to infect with a virus than cells that are actively dividing. So having a viral vector that can actively infect non-dividing cells efficiently is very important. And last, there's a low risk of genomic integration. So it cannot randomly integrate somewhere into the genome and, let's say, deactivate some tumor suppressor genes and thereby cause cancer. The risk with AAV is exceedingly low. What are the challenges? So I told you that it has a low immunogenicity, but it does not have no immunogenicity. So if you ever got into contact with AAV, chances are that you will have some antibodies that have formed over the years, and those antibodies may preclude you from being part in gene therapy trials. Those antibodies may inactivate the virus before it actually has an effect and introduces the gene into the heart. The other problem that we have is, yes, we know how to introduce a gene into the cell, but what do we do if we don't really like the effect that this gene has? Is there a way to turn it off? And right now, that is unfortunately not the case. We can't use AAVs for all different indications. So ARVC specifically for a gene called PKP2, plakophilin-2, works really well because the gene is relatively small. If you have a large gene that you need to introduce into the human heart, it may not work as well because AAVs have a rather small packaging capacity. We need to manage your caregiver's concern and manage expectations. And affordability is very important. So I told you before about spinal muscular atrophy. This is a deadly disease that affects children. They're born as floppy babies and don't develop normally. They never meet any developmental milestones. They're not able to roll over. They never walk. And usually by the age of 6 months or up to a year or two, it's a fatal disease. There is a cure, literally, for SMA now. It's called Zolgensma. It's a single injection of AAV. And it's remarkable to see those results. Nowadays, you see the kids who are actually going to school. You see that they develop relatively normally. You see them on their skateboard, so it's really amazing. But this comes with a price tag. So a single injection of that medication costs $2.1 million, and it's actually the most expensive medication in the United States at this time. So affordability will become a big issue. So how can we deliver the gene to the heart? Well, there are different AAV serotypes, and you can choose wisely there. So if you want to, for instance, transduce the brain, get a gene into the brain, you can use AAV2. If you're interested in transducing the heart, so in introducing a gene into the heart, you can choose a gene, or you can choose a serotype of AAV that has a tropism, as we call it, for the heart. Now, it's not 100% specific. Yes, it will go predominantly to the heart, but it will also go into the skeletal muscle. It will affect a little bit the retina. It will go into the lung. So we need something else other than choosing a viral serotype that has a tropism for the heart. And that's when we'll look at the expression cassette. What does it mean? So before we package a gene into a virus, it has to be in a certain vector where the gene expression of that gene is driven by what we call a promoter. The promoter is what turns the gene on or what turns it off. And you can choose a promoter that's active only in the heart. So if you combine a serotype of AAV that has a strong tendency to be expressed, to go to the heart, and then use an expression cassette with a cardiac-specific promoter and a gene that's expressed in the heart, there's a very good chance that you'll get predominantly expression of that gene in the heart. And last, you can decide how to administer the gene therapy. Do you want to go for a IV injection, where it's a simple infusion of the drug, and with the right AAV and with the right gene, it just enters promoter, it goes to the heart? Or you can also inject it directly into the heart to hope to need less virus, but achieve a higher level of that virus in the organ of interest, namely the heart in that case. So what about gene therapy trials for ARVC? So when we consider any kind of drug that's in development, importantly, before we come to humans, there is a preclinical development where we go through a very rigorous testing and say, yes, this drug is very effective, so it's worthwhile to start clinical research. There are several phases of clinical trials. The first trial is a phase 1 trial where we test a new drug on healthy volunteers. Phase 2 trials are, that drug is safe in healthy volunteers. Let's bring it to patients and see whether the drug has an effect, a positive effect. Does it do what it's intended to do? And last, if we see an effect there, we do what's called phase 3 clinical trial, where we confirm the benefit in a larger group of patients. So we start with a couple of patients, healthy volunteers, and then ultimately confirm the benefit in a larger group of patients. If the benefit is confirmed, this drug is usually marketed, and in the post-marketing period, it's phase 4 trials where there is ongoing real-world data, where there's still safety monitoring about the drug. Now, gene therapy trials are a little bit different, as phase 1 trials with healthy volunteers are usually not feasible. It wouldn't be ethical to give gene therapy to a healthy volunteer. So how do you do this? The way gene therapy trials are designed in ARVC is that early phase studies are combined. So they're focused on safety, that's the primary endpoint, but then we also look at efficacy, because all the gene therapy trials are designed in patients with a disease. And then later stages, to confirm the benefit in larger trials and real-world data, that all remains the same. So our top priority has to be safety. And there are some risks to gene therapy, there's no doubt about that. I told you the gene elicits, or the virus elicits an immune response is low if you give a little bit of virus, but with higher doses of the virus, you may see some immune response that leads to some inflammation, that may lead to changes in the heart, the liver, so they need to be very closely monitored, not only by cardiologists, but you need a dedicated team of gene therapy experts who are able to monitor and to recognize potential side effects early and treat them, because effective treatments are available. Another safety factor is that we start with the lowest possible dose. We'll make sure, again, safety first, we'll make sure that this is well tolerated. If there is no safety concerns with a low dose, then we escalate the dose, go to higher dose to improve the efficacy. And importantly, all those trials, there's an independent data safety monitoring board that reviews all possible side effects that may occur during the trial period. So who would be eligible for an ARVC gene therapy trial? And there are obviously different companies. They all have slightly different inclusion criteria, but I think this will summarize pretty well what is known so far, that it would be currently, it's intended only for adults, male or female. You have to meet the clinical diagnosis, the task force criteria. Patients have to have a defibrillator and have to be on stable medications for at least 30 days. The gene therapy trial that are currently in development are for PKP2 variants only. So that's the most common genetic cause for ARVC. And we're planning on including PKP2 patients only at this time. Importantly, about 30% of y'all will have had a prior infection or contact with AAV and will have developed antibodies. If you have antibodies against AAV, you are not a candidate at this time to be included in gene therapy trial. So we're testing for that specifically. It's important that contraception is used for 12 months, and you have to be willing and able to participate in long-term follow-up. And this is an important point. So I cannot go into the details about all the different trials that are currently ongoing because that information is not even publicly available. But I can tell you what similar trials in gene therapy will look like with respect to follow-up. So this is a study in Duchenne muscular dystrophy that is enrolling children. And as you can imagine, the bar in children is a little bit higher, so the follow-up is probably a little bit more intense here than what we would expect in adult ARVC trials. But it's expected that patients stay overnight as an inpatient for at least 7 days after receiving a single infusion of AAV. The length of study participation is also 5 to 6 years. So it's not like a single infusion, you go home, all is good, but you need close follow-up for a prolonged period of time. And now, how many follow-up visits are required? In this study, it's 49. So a lot of follow-up visits, and that's a huge commitment on part of the patients. Most of that is upfront, so in the first 6 months where there are a lot of follow-up visits, and later on the intervals are stretched out significantly, and ultimately you need a visit every 6 months for up to 4 years. And I would expect with some variability that adult ARVC trials in gene therapy will look similar. So who is not a candidate for ARVC gene therapy trial? Well, if your unwillingness to comply with study procedures, including follow-up. We recognize that that's a huge burden on patients, and this may not be for each and everyone. If you have severe heart failure symptoms, either severe right-sided heart failure or left-sided heart failure, that is an exclusion criteria too. If you're still actively participating in competitive sports, you're not a candidate for gene therapy trials. So we don't want to do this to allow you to be a competitive athlete. So we really want you to keep it light after you are included in these trials. If you have pre-existing significant liver or kidney disease, or if you have an active infection requiring systemic or antiviral or antibiotic therapy, you're not a candidate. So there are some open questions. So I told you that about 30 to 40% of patients who would be interested in gene therapy trials in ARVC may not be a candidate because of pre-existing antibodies. There are currently studies underway to see whether there is a way to get rid of those antibodies. Can we suppress the immune system sufficiently to allow patients to be enrolled in those trials? What is the optimal dosing of gene therapy? What kind of immunosuppressive regimen is necessary to minimize the immune response after you get the virus so that you have the best possible outcome? That is still an open question, and it's good that there are different companies who are exploring different options, and hopefully we'll get some good answers. What we also don't know is what is the optimal stage of the disease to do gene therapy. Probably not when you have virtually no symptoms, but you shouldn't also wait until you have very advanced disease. So somewhere in the middle is probably the best time frame. How long will the effect last? And if you see an effect that is beneficial, but the effect lasts for a couple of years. Will we be able to redose the patient who had an excellent effect in a couple of years, or will the autoantibodies that are generated by the AAV gene therapy, will that preclude patients to be enrolled in the future? So it's a very exciting time, and I think a new door has really opened to change the trajectory of the disease. And gene replacement therapy using AAV vectors is certainly a very promising new therapeutic options, which will likely transform the management of many inherited heart diseases, including ARVC. But currently, the only way to receive gene therapy is to participate in clinical trials. And I thank you for your attention, and I will be happy to take any questions later on. Thank you.